Okay, so we continue, okay, to the topics of these topics, ribosomes, okay? So you guys know about this ribosome. We keep on talking about ribosome. So what is a ribosome? So what form the ribosome? And we do know that the synthesis of the ribosome takes place inside our nucleolus. Okay, nucleolus is the one that synthesizes our ribosome, or more precisely, our nucleolus synthesize ribosomal RNA. So this ribosomal RNA actually from who? From nucleolus. Okay, from nucleolus. And this ribosomal RNA, okay, together with many protein form a complete ribosome. Okay, so if you want to classify your ribosome or to describe ribosome, ribosome can be found free in the cytoplasm, means that it does not attach to any surfaces. It's like floating at the cytoplasm, or they can bind or attach to the surface of the rough ER. Okay, so ribosome either can be found freely in the cytoplasm, means that no attachment at all on any surface like floating, or bound to the rough endoplasmic reticulum. But if you look at the structure of free ribosome and the Bowel ribosome, structurally they are identical and they can switch, okay, whether they want to attach or don't want to attach. So they are very freestyle in this case. So whether they attach or they do not attach, it depends on what kind of polypeptide they want to synthesize. So be careful when you answer the question for ribosome. What is the role of the ribosome? Okay, a general role, general of ribosome. Because when we learn about the polypeptide synthesis, ribosome have more detailed role. Okay, generally they synthesize. Highlight polypeptide. Okay, what is the role of ribosome? They synthesize polypeptide. Are you clear? They don't synthesize protein. Now be careful. Don't use the word synthesize protein. In order to form the complete protein, ribosome involved, RAR also involved. Are you clear? Because we want to fold the polypeptide into the precise three-dimensional shape. So that's why RAR also involved. So ribosome, the function is to synthesize, highlight, polypeptides. Okay, not proteins. Be careful when you use the word. Okay, so why they have the different form? Why they are freely located at the cytoplasm or bound? It depends on what kind of polypeptide they synthesize. If they are free ribosome, okay, free ribosome, they will synthesize the polypeptide later will be used within the cytosol, means that in the cytoplasm. So means that this is for internal use. Okay, so this kind of the polypeptides will fall inside the cytoplasm and then they'll be used within the cytoplasm. But if any polypeptide later will be used outside of the cells, for example, hormone, you want to send out hormone, you want to send out the enzyme, then we need to pack them, right? You want to send out something, you need to pack them. Correct or not? You try to imagine that, okay, if the one that you want to use it inside the cells, you don't need to pack them unless you want to use it for the lysosome. Okay, so basically the Bowel ribosome generally synthesize polypeptide. The Bowel ribosome, you want to insert the protein into the membrane, not internal use, at the membrane. You want to put the protein at the membrane, or you want to this protein will be used, okay, in the lysosome, or you want to export them from the cells through the secretions or exocytosis. So basically you use it, you want to pack them. If you want to pack them, then no choice, you have to use the Bowel ribosome to synthesize the polypeptide. Okay, now what is my insertion into the membrane? Now if this is our membrane, okay, for example, we need to have carrier protein. How we insert it into the carrier protein, how we insert it into the membrane? We need to form the vesicle. Are you clear? And then insert it. So what is the process? Now look at this. This is the membrane. It's something like exocytosis. This is a membrane. Then we pack we pack our transport protein onto our membranes of the vesicle. We pack them. So let them exocytosis take place, something like exocytosis. Then at the end, you can see that now we can pack them this way. Try to imagine. Exocytosis, and then with the protein. So when incorporate in already, then at the end, you'll form something like this. Wait, guys. So some parts you have extra membrane. Can you see that extra membrane? So this extra membrane will have the transport protein. Allow us to have the transportations. Okay, so this is how means that we mean the transport protein or carrier proteins that we want them to be incorporated or inserted into the membranes. Okay, so if you zoom into this ribosomal protein and RNA, they are arranged in the two very distinct ribosomal pieces of different size, which is large and small subunits. Okay, so this one very simple. I just disclose answer. You can see that they can join together, form a complete ribosome. Can you see that? When they carry out the polypeptide synthesis. When they are not carry out polypeptide synthesis, they will be dissociated into the large subunit and also small subunit. Are you clear? So they can dissociate. So we have the small subunit, large subunit. So they, when joined together, they can carry out the polypeptide synthesis. Okay, sorry, my mistake. Synthesis, not synthesize. Okay, so involved in the polypeptide synthesis, right? Synthesis. Okay, so what you can get here. So in this large subunit and small subunit, if I look at the detail of this large subunit and the detail of the small subunit, what you can get? You will get rRNA and together with protein. They form the, they form the ribosome. So ribosome later will synthesize the polypeptides. Okay, different protein, okay, always remember. Okay, we're going to learn this one next semester. Okay, now if you zoom into this ribosome, you look at this ribosome. The ribosome also have two forms in terms of their size. One is 20 nanometer, slightly smaller in the diameter, and another one is 25 nanometer. We call it ATS ribosome. Okay, so which one? If the prokaryotic ribosome, for example, bacterial ribosome will be smaller, 70S, and eukaryotic ribosome will be ATS. Means that those those cells that we learn with the organelle, okay, with the nucleus. This one means with nucleus. When they have nucleus, we term them as a eukaryotic cells. So these eukaryotic cells will have a slightly larger ribosome. So the ribosome, we term them as ATS ribosome. So what is doing ATS? So extra information, this one extra information is a Schubert unit. Schubert unit is how fast the sedimentations in the centrifugation process. It's not really a size totally, but it depends on how fast when we spin them, they will sink down. The larger the units, the faster they sink down. Are you clear? Okay. So basically, you try to imagine if this is the container. Okay, let's say we put in this our hot water, we put in our Milo powder. So Milo powder is so fine, they won't sink down immediately. So they take time for them to sink down. So how fast is sink down, okay, or we call sedimentation, okay, we give them a unit called Schubert unit. But generally, because our ribosome is too small, if you let them sink down by their own, they won't. Forever, they won't sink down. So we need to use a machine called centrifuge machines to spin them. When we spin them, they will sink down. We create a gravitational force. Okay, we create the gravitational force when we spin them. Okay, then we will be able to see, okay, this ribosome sink down. When it sink down, how fast? So it's basically the time. If the time is shorter, the S Schubert units will be higher. Are you clear? Extra information, sir. So the unit measure is the Schubert unit, a measurement of the rate of sedimentation. The faster the sedimentations, then the value will be higher. Okay, so prokaryotic cells 70S ribosome, eukaryotic cell 80S ribosome, and the ribosome also can be found inside the chloroplast and mitochondria, both of them 70S. Okay, now, don't worry about the small subunit and large subunit 30S and 50S. This one when you detach them. But for our syllabus, we only focus in terms of 70S and 80S ribosome. So to summarize here, we have 80S ribosome compared to 70S ribosome. So where we can get 80S ribosome? Cytoplasm of eukaryotic cell. 70S then will be cytoplasm of prokaryotic cells. Okay, mitochondria together with chloroplast. Okay, now sometimes question will trick you guys. Okay, how question trick you guys? Question will ask, in animal cells, where is the location of 70S ribosome? You have to be careful. You don't tell me chloroplast because animal cells don't have chloroplast. So 70S ribosome only can be found inside the mitochondria of the animal cells. Are you clear? But question say, if 80S, no problem, only in the cytoplasm of the eukaryotic cells. So means that if you look at this diagram, you know that's flectin sacs, kind of flectin sac, and with the dot. So what's the dot here? If you enlarge it, so those dot here, like this dot, right? They attach, right? So those are the Bowel ribosome. And you can see the dot actually floating. So those dot actually represents, okay, the ribosome, the free ribosome. But we do know that it's at the cytoplasm, right? So those ribosome write down not only the normal not only ribosome, but in fact they are ATS ribosome, right? They are ATS ribosome. Okay. So with this, I have done for the ribosome.